Well, listeners, welcome to our next edition of Science of the Times podcast. And I think, Simon, we've both been in Oxford all week. First time that's happened for a while.
It is actually just thinking back to the last time. Yeah, and both of us at the same time. So maybe this is the sort of, out of term advantage.
Yeah, we're both doing it remotely.
Yeah, true.
We,
We're a few
well,
meters apart physically, but
no, exactly. Well, so today, I mean, this is kind of really your closer to your expert area of expertise than mine, apart from snakes.
yes. Yeah, I'm super excited about today's guests. So they've been working on developing proteins, and here's the key, starting from completely new designs and scaffolds of protein, just using computational methods to neutralize the venoms of snakes.
So what's this? Sorry, let's start. A protein. We talked about this before, I think, with when we were talking to the Oxford nanopore
Yeah.
people. These are the workhorses of life, and the structure
Yeah.
is really important. So,
Yeah, so it turns out that the structure is important, and if we know the structure, we can learn quite a lot about how they work. And it turns out that now we can get the structures and predict the structures of loads of these proteins.
and you use the word scaffold. So, what's a scaffold? Is it like scaffolding we put outside our house?
It's not too different, actually. So it's just saying that at the sort of underlying, if you like, skeleton, as it almost were, of the protein that holds it together and gives it the sort of you can also think of it as a backbone, or the equivalent in humans, if you like, of the skeleton. And then on top of that you'd have the flesh and all the extra bits, which would be the amino acids. And I think the key is that we can use X-rays, we can use microscopy and other images, and now, thanks to advancements in artificial intelligence, we can also predict the structures of these proteins, even if we don't have the actual X-rays or images. And in fact, two of the recipients of the Nobel Prize in Chemistry this year, John Jumper and Denis Hassabis, were given the Nobel Prize for these advancements in being able to predict the structures.
And so the reason the structure is important is sort of proteins talk to one another by joining together, and the structure allows them to fit together. A bit like I don't know, fingers coming together or bits of Lego coming together and sticking together. That's how they work, isn't it?
Yeah, absolutely. And the other thing is that it's not just proteins that can stick together. Other molecules can stick to the proteins, and then they can make things happen. So, for example, in the case that when we talk to our guests later, you can have, for example, a protein that some other molecule comes and binds to it, so they talk, and then that protein changes its shape, and that sends a signal to the next protein. That change sends a signal to the next protein, and then things can happen, like we move our muscles, or we have any process at all that occurs in the body happens because these proteins are talking to other molecules and to each other.
And what our guests have done today, I think, is they've basically found a way of gluing up one of those sites so it stops that chain of events happening. Is that right?
That's right, and they've done that using again computational methods. So the interesting thing here is they've designed these proteins. They're not so whilst we know what the structures of real natural proteins look like, what happens if you want to design a completely new one that nature or biology hasn't thought of? And you want to, it's almost humans sort of playing God, if you like. And the techniques that have enabled this were developed in the lab of David Baker, who was the third recipient of the Nobel Prize in Chemistry. And it was for this way of designing completely new proteins that nature hasn't come up with.
So these are molecules that the world or the universe may never have seen before.
That's right. And
Amazing.
um, and then not only do you design them and make them, then you have to find a way to continuously make them if they're to be useful. And I think our guests will tell us a little bit about how they do that with their designed proteins too.
And going back to snakes, they've been focusing on the snake venom,
Yes.
and they're basically making these terrifying snakes you get over in Australia called elapids. just like these kind of nice, cuddly things we can pet because if they bite us, it doesn't matter.
You go first. Let me know if it doesn't matter, and then I'll give it a go.
Should we
Yeah.
call them in and let's
Yeah,
chat to
let's
them?
do that. Okay, great. So, Tim, shall we let our guests introduce themselves and then we'll get started? So, over to you, Tim.
Great. Yeah. So thank you very much for having me here today. My name's Tim, and I'm an associate professor at DTU Bioengineering. That's the Technical University of Denmark, and run a research group focused on protein design with a historical focus on snake bite. And that's also how I got into this particular paper that we're discussing today.
Hello, my name is I'm Susana. I'm a recent grad student from the Baker Lab. Right now, I join a postdoc position, and yeah, I work in David's group, which is like a world leader in protein design. So I guess my role was in the more design part of the project.
Okay, great. So, you were a PhD student in the lab and now you're a postdoc, is that right?
Exactly. Yeah, I was a PhD while the paper was being done. Yeah.
Okay, and actually, I was just thinking before we go into the science, so the paper was being written before the Nobel Prize was announced? Was that so just trying to get a feel for what happened at that time and the sort of excitement, I guess. Can you tell us a bit about that?
Yeah, it was written before. So I think, yeah, we started the project on May last year, and David got Nobel Prize like by November, on the same year. Is that correct? I think so. Yeah, I think that, regardless, the Nobel Prize came after the project.
And did things change in the lab when the Nobel Prize came?
I mean, when I was there, I don't think so. I think when the Nobel Prize was announced, everyone was very happy and we had like a very, very big celebration. But next day, everything was back to normal. Like David was making his salad for lunch, having meetings with his students and postdocs, and nothing really changed in my perspective. And as far as I know, from my colleagues who are still there, everything is the same.
The brutal nature of science, right?
What would you do, Syma, when you win a Nobel Prize? Will you make everyone in your lab curtsy or bow to you?
Well, both, one by one, and then I'll move to the Caribbean and buy a great big yacht. But how about you?
Oh, God, no, I haven't even thought about it. I'd probably retire.
Now you're making people think that I've thought about what happens. I think about this all the time if I win a Nobel Prize. Anyway, shall we dive into the science? So, yeah, this remarkable paper where you've designed de novo a protein that can neutralize snake toxins. So I think it's interesting for me and Tim, and I think our readers, first of all, to perhaps understand a little bit about how you got into snake toxins first and foremost.
I mean, for me, the journey started very young. When I was about 4, we went to Indonesia. I got to hold my first snake, and that was a life-changing moment. I got fascinated with the animals. Then at 6, I went to Australia, declared to my mom that I would study venomous animals when I grow up. And she laughed at me and tapped on my shoulder and said, "Sure, son, sure." And then, yeah, 12 years later, I moved to Australia, studied venomous animals, and then got more and more interested in the therapeutic angle rather than the zoological angle, and started becoming interested in how these envenomings are being treated right now, which is quite crude. It's using basically purified horse blood, and thinking about how we could maybe come up with new and ideally better and cheaper solutions that could go to those most in need. And that's where my journey took me to the UK, and then last but not least to Copenhagen to try and work on different solutions that could go into that space.
So did you wander around in Australia trying to capture things like their brown snakes and their death adders and their inland taipans?
Definitely, definitely. Yeah, I had a lot of fun close encounters. I was also at the age where you not necessarily make the smartest life choices, but I came out unscarred overall. I still have all of my fingers, so that's also nice.
Yeah, excellent. And then so Susana, and then you joined the team. I guess you're in the Baker group, and you're obviously in a group that's working on protein design and has been established in that area for many years. So how did the team assemble? Like, how did you and Tim get together and decide that, this is what you were going to work on?
Yeah, I mean, yeah, that's very interesting because I was in the last year of my PhD, and the protein design methods like were working very well. And then David told me like, Susana, just have fun, like think of something that will be fun and use protein design for that, right? So I spent a couple of months like reading about like how protein design could be useful in some therapeutic area, and I came across a lot of papers where they say snake bite is something that is really urgent and we need better therapies for that. And then I kind of like was having a coffee in the IPD with some people, and then this guy Joe Watson, who was a postdoc member at the lab, who is the main author of the Rosetta Fold diffusion paper, like overheard my conversation, and he's like, "Wait, Susana, there is like this professor in Dinamarca. His name is Tim, and he just like sent me an email months ago, and he wants to do exactly the same. But I'm so busy, and why don't you talk to him?" So I think I just wrote an email to Tim, and I think that's how things got started, right? Like it was like it came like in the perfect timing, and I think Tim and his colleagues were thinking about this, and I was also at the same time thinking about the same thing, and we just converge into the same space, I guess.
I guess like so much of science, right? So much of it is there's a large element that's serendipity and just timing and things
Yeah.
work out. Tim, did you already have a set of proteins in mind that you were working on or scaffolds, or was this something where you decided to say, okay, we're going to start completely from the beginning?
I think like the whole project, everything was a very collaborative process. So we had some ideas, Susana had some ideas. We discussed which ideas we should pursue first, and then we gave it a crack and saw what would make sense. But we landed on the logic of going for some of the most lethal components of snake venoms, these three-finger toxins. And the reason for that is one, their lethality, and also because current antivenoms are often not particularly good at neutralizing these toxins because they're very small. And the way that we are producing antivenoms right now, we need an immune system to recognize these toxins to then produce antibodies, and they're not very recognizable by the immune system. So we decided that would be a good choice, and Susana got designing and did some amazing work there and getting some cool candidates that we could take further.
So can I take a step back as being the sort of non-specialist here? So snake poisons, it's a protein, right? When they bite you, they're injecting various compounds into you, but the three-fingered ones you're talking about are a protein, is that right?
Yeah, so snake venom is basically all proteins, and I'll correct you here. It's venom, not poison. The beauty of the English language is that there is actually a distinction. Many other languages don't have this distinction, but in English we do have it, and the distinction is primarily based on poisons being small molecules and venoms being proteins. And that's also why snakes need to inject their venom because they don't absorb through the skin like small molecules would. Whereas your poison dart frogs, they have these small molecules that you absorb through your skin. So yeah, but long answer short, these were proteins, and snake venom is a protein cocktail of different things that modulate the immune system, that modulate your cells, and by modulate I mean mostly destroy. So they have this whole arsenal of different biochemical weapons that they deploy, and we focus on one of the most lethal groups there.
And so the snake injects it into the muscle or into the blood, and is it attacking blood or is it attacking nerve cells? Is it neurotoxic or hemotoxic?
So snakes can have tens of different toxins that all do different things, right? So, and where they inject them also depends a bit on where you get bit, how long the fangs are of the particular snake. So most of them go into your lymphatic system, so just a bit more shallow than hitting the blood vessels. You're very unlucky if they hit the blood vessels, because then they'll spread very fast.
Quickly.
But yeah, in terms of what they do, it depends on the species. But many have this multifactorial approach to trying to subdue their prey, so paralyze them, digest them already, pre-digest with the venom, give them heart failure so they can't run away. Different reasons, but it's all for prey capture, right?
And then,
So
so
Tim,
what
the
you,
one
sorry,
thing.
what you did, just so I've got it clear, and so our listeners got it. You have then designed, using computers, you've designed something that neutralizes this one particular class of three-fingered protein. Yeah,
That's exactly what Susana did.
amazing. So cool. Sorry, I just wanted to get that so I understood, Syma.
Yeah, no, that makes sense. So in terms of the protein that the poison binds to, so in this particular, sorry, the venom binds to. I'm a fast learner. So it's so this protein, this receptor is responsible for sending signals to, for example, muscles that then mean they contract. And so if you interfere with that protein, then the brain is sort of saying move, but then the signal doesn't get to the muscle and the muscle cannot contract, and so you get some sort of muscular paralysis. Is that correct?
That is what two of the types of toxins that Susana targeted do exactly. So we prevent them from blocking to a receptor where these muscle transmissions usually get either activated or deactivated by these toxins. But there's another one which we also targeted that uses the same structure but actually attaches to membranes and disrupts cell membranes to kill cells. So that's cytotoxic three-finger toxins that we also targeted.
Very close to my heart, disruption of membranes, but I could talk about it for ages. So, Susanna, so do you want to talk us through? I mean, it's difficult for me to, and I'm as you know, a computational biologist, but really difficult for me to understand where you even get started. So you have this protein, and I think the traditional approach is to target the receptor protein, but what you guys did is you targeted the venom itself to try to stop it from binding. Is that right?
Yeah, that's correct. I think we try to follow this approach as it seems like from the structural point of view, when you look at the toxins, they have multi-stranded structure. So I don't know, just like for me, when I saw that, I felt like it could be easy to feed like another protein that could match in shape with that toxin and just make it very bulky so it cannot longer bind to its native receptors. Yeah.
So, okay, I'm just trying to understand. So if we think about it a little bit like if the protein, the receptor is a little bit like a socket, and then you put a plug into it, and then the power goes and something happens and the muscle moves. So what you're trying to do is you're trying to prevent the wrong plug, as it were, from going in or something. And so you just block it off. You put something big, ugly on top of it so it no longer fits into the socket.
Yeah, that's correct. Yeah, I wouldn't say it's ugly, but yeah, it just the protein.
From a molecular way.
So it's molecular Lego.
Yeah,
Yes.
yeah, exactly.
Okay.
Yeah.
And can you talk us through the techniques you use to do that? Because I mean, I'm a computational person, but I wouldn't be able to do that. So what's the sort of specialist knowledge that you guys use to start designing these proteins?
Yeah, I mean, we use like these kind of like tools, protein design tools that have been developed like before our paper that are very powerful. So the first tool that we use is called Rosetta fold diffusion. So in this method, it's actually based on image reconstructing method. So basically, it create protein shapes from like noise. So basically, you can specify to these models which are very powerful and have, I mean, they don't know many things about biochemistry, but they know how to build proteins around other proteins. So you basically provide a toxin structure, and this model generates a lot of solutions around this toxin. So try to match some shape that would bind to this toxin. And then this is like a backbone level. So we don't have any amino acid sequence. We just develop like a Lego that fits into another protein.
So a scaffold, basically.
Exactly, like a scaffold. So this is very computer efficient and very fast compared to previous methods before deep learning. Then we use another tool that was also developed by the Baker Lab, which is called protein MPNN, which basically gives an identity to this backbone. So it gives amino acid sequences that falls into the same backbone, right? So you put different amino acid sequences into them, which is also good because you have different amino acid sequences that could potentially make different interactions with the target. And then in the last step, we use AlphaFold or Rosetta Fold, which are a structure prediction method, just to make sure that the complex between our target toxin and our protein that we designed actually like are fitting together the way we designed them. So this is like an oracle that enable us to filter what is good. So we have like some computational metrics that tell us this is likely to bind, this is not likely to bind. So that allow us to reduce the number of proteins we test in the lab. And because these tools are so great, it works very well.
Did it
Okay, so
work?
let
Did
me.
it
Sorry.
work first time?
Yes.
Wow, that's amazing.
Yes.
So I'm just
Yes.
trying to understand what you said about the lot of complexity. So you have this scaffold first. So you just take the backbones. We don't worry about the amino acids. So taking Tim's analogy of the Lego, so we just take one type of Lego brick and we build a scaffold, and then you put in the amino acids. So then you try putting in extra different bricks in different places to try to improve the complementarity, or did I get that wrong?
No, in the second step, so at the first step, you only have like a backbone level. So everything you want to do, like about making different like contacts,
Yeah.
is like kind of settled there. And in the second step, you only give an amino acid sequence. So you don't add any extra proteins. Like you build contacts based on the amino acid identity.
Sure, I just meant extra bricks. The extra bricks are the amino
Ah, the
acids.
amino acids. Ah, okay. Yeah. Okay. So then you're
Okay,
right. Yeah.
so that's all computational and it's all sort of theoretical, as it were. But then you have to use AlphaFold or Rosetta to see what would it actually look like, and does it seem plausible? Is that right?
Exactly. That's exactly what we did. And I mean, these tools are so powerful that could be translated to any kind of problem. Snake toxins is one example, but you can use the same pipeline for different applications. And the beauty is that they work so well that even in our first experiments, we got proteins that bind to toxins.
Great. And how do you take it out from the sort of computational world, the theoretical world, and test this? Sort of what are the stages involved in testing it in in the wild, as it were?
I mean, because we basically in the protein design process, you end up with amino acid sequences that fall into specific structures. You can just take an amino acid sequence and get the DNA, and you can order those DNA sequences from companies that synthesize DNA, and we can buy them and put them into bacteria that we use as a factory to produce our proteins, and then we bring to life these theoretical proteins, and we test if they do what we made them for.
So you use the poor little bacteria to make these proteins that they were never ever designed to make. In fact, nature never designed them. You designed them completely from scratch, and then you have the bacteria working like little factories for you producing these.
Exactly. That's exactly what we did.
Excellent.
That's really amazing. I mean, it's so and so you use quite a lot of machine learning and artificial intelligence along these steps, do you?
Yeah. Yeah, all of these steps, all of the pipeline or the toolkit that I described is based on deep learning tools.
Yeah, so
All
you
of them.
couldn't have done this until sort of 5 years ago. I mean, it's
No. I think also in the Baker Lab was a huge, like a huge change before these deep learning methods appear. Like a project like this would have taken, I don't know, 6 years or so. But because these methods are so fast and so efficient, it took us a year, which is much more faster.
wow.
So really, like deep learning came to really change everything, and also the fact that we have extremely powerful oracles makes a whole difference, because in the past we didn't know what was good and what was bad, but right now we almost sure what is good and what is bad.
And then you sent these blockers, these new design proteins you've got to a lab, and the venom was injected into mice, and then the blockers were, is that right? Did I get that correct as well? So that you actually then tested it in mice and it worked.
Yeah, as Tim said, it was like a collaborative effort. So we
Yeah.
designed these proteins, we test them in the lab at like in vitro, not very realistic. Like in test tube, you have like some biophysics assays that makes you think if it binds or not. And then for the real world application, a lot of experiments were done at DTU in Denmark, but also at the University of Northern Colorado with experts that know how to perform these experiments in animals. So we kind of split the work across the globe with different scientists. Yeah.
And Tim, can I ask you a question then? So, is the solution that Susana and the Baker team found here what you would see in naturally occurring antivenoms in animals that don't suffer too much from these snake bites? So, for example, some of the Australian marsupials are quite resistant to elapid snake bites.
No, not at all. It's quite different. I mean, most animals try to avoid envenomation or minimize the effects by actually changing their receptors to these neurotoxins. So they have minor changes in the receptors themselves, so evolving away from the native state, so the toxins cannot bind to these receptors anymore. That's the major thing that animals try to employ. Some of them have decoys, but most of them just try and evolve away, and that's been this evolutionary arms race of making these toxins more broadly neutralizing and so. So, yeah, it's unfortunately not a solution we can use in humans. Gene editing is slightly frowned upon still, so we pursued a different therapeutic avenue here. And also, one fun fact I think is that the tools that Susana's group or David's group at the University of Washington have developed have really built on major advances in completely different areas where machine learning has really boomed. And it's just a great example of how we take things that are great for language, right? These chatbots, ChatGPT, they're based on these transformers. That's what made AlphaFold great. People built this for different purposes as well and used them a lot in other areas, and we've kind of ported them over into the biosciences now. And the same with the thing that made the generative process, the design of these proteins so brilliant. The machine learning architecture actually comes from image AI, like Midjourney and DALL-E 2, where you say, I want a monkey riding a bike on the surface of the moon, and it generates this. Same architecture was brilliantly applied by Joe Watson, who Susanna mentioned before, and the whole team at the Baker Lab to transport this concept into the protein world.
And so are these sort of pipelines that you used here now, are they ready to go and deployable for a whole range of different venoms? And I guess could be anything where you want to neutralize any kind of, any protein at all. So I guess these sort of almost complementary proteins now can be designed. Is that right?
Yeah, absolutely.
And so do we think this is a sort of a transformative, it's like a step change in the way we're doing drug discovery, I guess. Is that, or is that overblowing it, or do you think that's right?
I think it's one of the most transformative technologies I've seen in my lifetime. It's also, I mean, you don't get a Nobel Prize for anything. And I think particularly the latest progress, I mean, obviously there's been a strong history of progress in the field by the Baker Lab, but especially the latest has really changed the way that we do science and also drug discovery. And I mean, Susana can probably add a couple of examples that she's also worked on and others in the group, but it's completely game changing, going from a discovery phase that takes maybe a year traditionally to a month using these AI tools. So you can really speed up response times. Let's say we have another pandemic and we need these proteins as reagents for new diagnostic kits so we can track the pandemics, or even better, as therapeutics against them, right? The time is everything, and these tools give us a lot of time.
So what next, Susana? What are you working on now then? What's the next thing you're gonna crack?
I mean, right now, as I mentioned, I joined a position at a postdoc position in Madrid. It's as a national research cancer center. So, I mean, we're working with. I would consider myself a protein designer. I'm more like a Lego builder, like an engineer, not like a very biologist. And I think cancer is probably one of the hardest topics anyone can study. So, I think I would like to use these protein design tools to unravel like very complex things about cancer and hopefully design a better and more accessible therapeutic options for cancer patients.
And what are the downsides of this? So could it be used, and I've been watching too much sci-fi, could it be used by for malicious causes, sort of chemical warfare? Is that something that what are the ethics of doing this?
Yeah, I mean, I can just maybe a team can build on this, but I think that's definitely a big concern. And I think recently, I mean, I know especially from the Baker Lab that that's one of the biggest concerns, that there have been big discussions on how to regulate, like these tools, because right now all of these tools they're open source, so the code is available and technically anyone in the world can run them. Even right now, if you wanted to try it, you could just open your Google Chrome and run a protein design, right?
I assure you, he wouldn't be able to.
Well, that's what I'm going to do this afternoon.
Yeah, they're like it's very automatic right now to use these tools. So I think we need to to make like new kind of like rules, or yeah, like new kind of rules, like how can we use these tools in a responsible way to avoid malicious people to use them, even like regulate like how you order genes, how you obtain the DNA. Maybe you can design that stuff, but if you cannot buy the DNA, that could potentially reduce the risk. I think the governments need to be involved, and the scientists should stop focusing a little bit less in science and also more on ethics and how can we use this more responsibly.
I guess no one really anticipated the wider impacts of AI and how it would transform almost every aspect of science, right? So it's almost a whole new field now, the ethics of AI in itself. Because when you were first talking about the complementary shape to the venom, I was thinking, well, what stops someone designing something that actually will cause the paralysis?
Exactly.
So yeah, it's a frightening thought.
So
I mean,
sorry,
just jump in on this conversation. So I think again, it's chemical warfare and bio warfare are two very different things, and proteins are not necessarily the best thing to use if you really wanted to have a broad scale impact. For targeted malicious intent, it's great, but again, as we talked about, it's hard to get these proteins into the human body.
Yeah.
You can design them along other organisms and so on that can help with that, but it's not the most straightforward way to create, I'd say, weapons of mass destruction. But having said that, obviously, I fully agree with Susana, and I've also talked to several government agencies here in Europe to advise a bit on this topic, because you need to be prepared, and these things are moving so fast that you'd rather stay on top of it. And ethics have been overlooked in AI in all spaces, exactly like Susana said, and there needs to be that focus. And luckily, people who are major leaders in the field, like David and others, are also putting a bit of a lens on that. So people are paying attention, and government agencies are largely paying attention.
So why did you start with snake? Let's go back to the paper very briefly. We're running out of time, I realize, but why did you start with these three-fingered snake venoms? Was it because you thought that they had a particular easy structure to crack, or is it really because you just love brown snakes?
No, I mean, as discussed earlier, the two main reasons were, one, that they're super important components of snake venoms. They are very, very toxic, and current solutions aren't working very well against them largely. So while we might still be some steps away from having a fully fledged de novo designed antivenom that covers whole areas like sub-Saharan Africa, we were envisioning that maybe at least these compounds could be used to reinforce existing antivenoms and just be an additional component to existing treatments while we're working on broadly covering regional antivenoms that are multi-species and so on and so forth. So that was some of the thinking that we put into this.
Just the structures of the venom proteins that you went for, was it also particularly because they had these three sort of protruding loops and you thought these would make a good target, or was that just you picked them and then you saw the loops afterwards?
Susana might disagree, but I'd argue that they're not some of the easiest toxins we could have gone for, because they have quite a lot of flexible regions still, and they are not very big, so there's not a lot of contact space. There's like for instance, phospholipases, which are also pretty nasty. You have a lot more options to work with. So I wouldn't say this was an easy case scenario, but I don't know. What do you think, Susana?
I think, yeah, definitely maybe not the easiest target. For me, I was looking into them more like, as I said, like Legos, like they have like a lot of beta strands. And I thought like, why don't we make complementary beta strands into them? So I was looking more at the structural level. And I thought like, if we make very high affinity interactions with high specificity, we should just block their function regardless of their flexible loops, or I thought that should be enough, and it turned out to be enough.
I have to say it's amazing. I'm really, really impressed. I have to admit, when I was at university, which was a long time ago, the kind of structure of proteins and things didn't really grip my attention. But now hearing what you say, it's just fantastic. I mean, many congratulations to you and to everyone on the paper. It really is quite a remarkable achievement.
I think especially going from computer design to
Yeah.
in vitro testing to in vivo, that whole, all the way through from a concept to something that actually works is is amazing. And in a year too, that's for our listeners and viewers, that just to sort of reinforce that's remarkable. Most therapies take many years to make, and with a lot more sort of investment than you guys have put in terms of the resource. I think you'd agree with saying, right? It's been efficient.
Yeah, I mean, I think the speed is amazing, but I also need to point out that it's, first of all, a collaborative effort, and also we had a lot of resources in terms of computational resources. The Baker Lab has immense computer power where I can run all of these design campaigns much more faster compared to other smaller places. And we also have a huge capacity to order DNA, which is something I would like to mention. Just to, I mean, I think you could do this at smaller scale, but I want to mention that because it's important also to
Absolutely.
say this.
That access to computing is, without that, I think you can't get started, right?
Maybe it would take 2 years in a smaller place, but we had access to great collaborators, access to great resources, so that's something very important for this speed.
Of course.
And I think if I may add, Susana is also maybe underselling herself a bit there. She was working day and night, and incredible. Like the speed at which she was generating data was mind blowing. So I think having the right people on the project, not just the collaborators, also the main driver here, makes a big difference in getting these things pushed forward. But technologically speaking, it is definitely something that is a paradigm shift in the field.
Yep.
So I think we've about run out of time, Syma, but so thank you so much for taking the time to come and talk to us. It's great getting into, it's great to have talked to members of such an international team, and we've got, Denmark and Spain represented here as countries. So it's been really wonderful talking to you, and good luck with the next challenges. And for our listeners, as you go down to subscribe, also you'll see a link to the paper so people can go and read the Nature paper. But thank you so much.
Thanks, guys, and stay away from those snakes, Tim.
Will do my best. Thanks for having us.
Okay.
Thank you so much for the invitation. Bye.
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 Well, that was amazing, Syma. I'm kind of, just blown away that you can take a shape of a protein that causes harm, and you can then on the computer design a solution, something that's going to bind or block the protein doing its job. Then you can make it, and it then works when you actually do it. And it's kind of involved computers, it's involved bacteria, it's involved snakes producing the venom, it's involved mice. I mean, this is a deep understanding of biology, isn't it?
It is, but I really liked what Susana said in that she thinks about it like an engineer. So it's also a new way of thinking about biology, also thinking about the underlying shapes, and almost perhaps a bit like an engineer and an architect, if that makes sense.
No, it totally makes sense. I mean, this is, as you say, it's like designing a completely new molecule with a shape to do a particular job. And I think that's just remarkable.
And what the scope of it can be in the future is mind-blowing. Susana mentioned cancer that she's working on now, but I was thinking also about, misfolded proteins are a huge problem, as we know, in neurodegenerative diseases. So you can imagine it could be revolutionary because those proteins are very hard to target.
Well, even things like mad cow disease is it's a prion, isn't it? Which is a misfolded protein.
Yeah.
So maybe you could gum those up and stop them working.
Yeah, absolutely. Yes, just another way of thinking about it. Design something that actually just neutralizes them and stops them doing nothing. I think it'd be amazing.
No, as Tim said, this is just an astonishing bit of biotechnology. I mean, it has the opportunity to revolutionize all sorts of disease treatment, inflammation, I imagine, as you said, cancer. Because a lot of diseases, a lot of genetic diseases are because of proteins aren't working properly, isn't it? So So
Yeah,
I guess,
absolutely. But I guess, we think about the positives, but then, they also touched on the potential negatives too. There's a huge ethical concern and a risk that these things could be manipulated for, nefarious reasons, I think.
although Tim did have a good point, getting them into the human body isn't straightforward. So I
Yeah.
completely agree we have to focus on the ethics, but maybe there's a saving grace there that makes it hard, to mass infect people with proteins that don't work very well.
Yes, hopefully. I think, yeah, you're right. I mean, they're too big and bulky, aren't they, to get in. So that's a saving grace.
But again, on our kind of journey, here's a case where machine learning is cropping up, allowing us to do things we couldn't have dreamt of doing just even 3 or 4 years ago. I mean, it's remarkable, isn't it?
Yeah, I mean, and to think this technology is everywhere. Tim mentioned Midjourney and Dali and these things that we use all the time now. And even making the logo for our podcast, we use the Photoshop generative AI tools, for example. Things like that. That those tools can now also be used to design proteins.
Yeah.
Uh, and where else are they being used? It's mind-boggling.
Well, in fact, that's so in 2 weeks' time, our guest is going to be Professor Chris Summerfield, and he is an expert on he's a neuroscientist, but an expert on large language models. And I'm really looking forward to talking to him because I think he'll be able to give us some ideas of where else we might be able to expect these things cropping up. Anyway, excellent. Okay. Bye-bye.
Bye.